The collective composition of trace elements in the human body is known as the metallome. While genetics receive the bulk of research attention, the precise balance of these essential elemental micronutrients plays a critical, yet historically underexplored role in the pathophysiology of autism spectrum disorder. These scatter plots show the results of inductively coupled plasma mass spectrometry profiling from human plasma, establishing an exact inventory of 21 different trace elements across both a neurotypical control group and an ASD cohort. When we focus specifically on copper, the data reveals a stark, significant reduction in plasma concentration within the ASD cohort compared to developing controls. While copper levels plummet, the concentrations of other critical elements like selenium, rubidium, and strontium remain identical to neurotypical controls. The data indicates a targeted elemental failure rather than a systemic nutritional collapse. Plotting these copper concentrations against the Autism Diagnostic Observation Schedule, or ADOS-2, establishes the clinical stakes. The data yields a strict inverse relationship. As plasma copper drops, the severity of social impairment and total symptom scores rise. These T1-weighted MRI scans transition the focus from blood plasma to macroscopic brain morphology. The highlighted regions track the density of white matter, the heavily myelinated nerve networks that connect distinct areas of the brain. This box plot quantifies the physical anatomy. Individuals within the ASD cohort showed a significant, measurable decrease in both total and frontal lobe white matter volume. And mapping that white matter volume directly against the clinical symptom scores yields the exact same negative correlation we saw with the copper deficit. Reduced white matter tracks specifically with worsened social interaction and social affect. The clinical data presents a clean chain of evidence. A specific systemic trace element imbalance in human patients directly correlates with reduced macroscopic white matter architecture, which in turn maps exactly to complex behavioral phenotypes. To prove this correlation is driven by an actual biological mechanism, researchers need to generate a targeted copper-deficient animal model. They achieved this by administering TETA, a selective copper chelator, to mice during embryonic development. TETA binds to and removes copper from the system without disrupting structurally similar periodic elements like zinc. These heat maps map the movement of the offspring during a standard 3-chamber social interaction test. The control mice reliably cluster around the novel social target, indicated by the dense red zones. The accompanying bar charts confirm that the TETA offspring exhibit severe, highly specific deficits. Their interaction time and total distance traveled around the novel social target plummet compared to the control group. The marble burying and grooming data capture another core phenotype. The copper-deficient mice exhibit significantly higher event counts in both categories, proving the presence of restricted, repetitive behaviors. These fluorescent microscopy images of the prefrontal cortex identify the physical source. Markers RXRG and ADCY5 highlight oligodendrocyte cells, which generate white matter. Comparing control to tetra, the stark absence of these cells links copper deficiency directly to stall development. The quantification panels below show how this molecular failure manifests physically. The populations of early progenitor cells and fully mature APC-positive oligodendrocytes have collapsed, resulting in severe hypomyelination throughout the cortex. The tetra mouse accurately replicates the human clinical presentation, providing a validated biological baseline to hunt for the exact cellular trigger of this neurodevelopmental failure. To locate the exact moment this structural defect begins, we must rewind the timeline from adult hypomyelination back to embryonic day 11.5, isolating the tissue. During this highly active window of neurodevelopment, robust microvessel formation, known as angiogenesis, is an absolute requirement to deliver oxygen and nutrients to rapidly dividing cells. These fluorescent images use lectin markers to map the vascular network at E11.5. In control tissue, branching microvessels are clearly defined, but copper-deficient TETA embryos display severely reduced vessel branching. Quantified data points to a steep drop in lectin-positive area and microvessel density. Despite this severe vascular defect, the overall population of neural progenitor cells, identified by markers like PAX6, remains completely unchanged. The tissue has not undergone generalized cell death. Instead, this highly specific failure of microvessel formation plunges the developing embryonic brain into a dangerous hyper-hypoxic environment, starving the tissue of oxygen. This flowchart models the biological failsafe for hypoxia. Cells upregulate HIF-1 alpha, which binds to DNA to transcribe genes like VEGF-A and GLUT1. In teta embryos, HIF-1 alpha spikes, but missing its copper cofactor, the pathway stalls before initiating gene expression. The immediate consequence is a failure to transcribe those critical downstream targets. The tissue cannot produce VEGF-A to build the vessels it needs, nor GLUT1 to process glucose. This triggers a metabolic crash. Without sufficient glycolysis to produce energy, the cellular mitochondria are pushed beyond their functional limits. They begin leaking massive amounts of reactive oxygen species. These images capture physical evidence of this toxic state. Red fluorescence tracks 4-HNE, a biomarker for oxidatively damaged lipids. Notice the high-intensity overlap below. Severe oxidative stress concentrates in the ventricular zone, mapping directly over OLIG2-positive cells, the exact progenitors building oligodendrocytes. By stalling the hypoxia failsafe, copper deficiency forces the mitochondria to actively poison the local progenitor environment through unchecked oxidative stress. When subjected to this profound oxidative stress, the oligodendrocyte progenitor cells execute a specific defensive response to survive. The red channel here isolates the mitophagy factor BNIP3. The data reveals a massive upregulation of BNIP3, strictly confined within the OLIG2-positive progenitor populations of the medial ganglionic eminence. BNIP3 mediates mitophagy, the autophagic destruction of failing mitochondria. BNIP3 flags the damaged organelle, and an autophagosome engulfs it. An autolysosome degrades the isolated mitochondria. This prevents outright cell death, but simultaneously triggers an emergency signal to the nucleus, forcing cell cycle arrest. The genetic consequence of this shutdown is absolute. The critical proliferation markers HES5 and ID2/4, which normally drive the expansion of the progenitor pool, are immediately suppressed. This forced mitophagy acts as an irreversible cellular break. It permanently depletes the progenitor pool, erasing the cells required to generate adult brain myelination. The massive induction of BNIP3 and the resulting metabolic stress converge on a single downstream target, the mTOR signaling pathway. This network is the primary chemical engine for oligodendrocyte development, and in these copper-deficient cells, its activity is severely suppressed. At the exact same time, the copper deficiency actively upregulates DDIT4 and EGR1. Both genes act as redundant biological suppressors, ensuring the mTOR pathway is completely locked down. This presents a unique opportunity to test the mechanism. If mTOR suppression is the terminal cause of the white matter defect, artificially forcing mTOR activation should bypass the original embryonic trace element deficit entirely. To test this, researchers administered a direct pharmacological mTOR activator. They used MHY1485, a synthesized compound designed to forcibly reactivate the entire signaling chain. This Western blot analysis provides biochemical proof of concept, tracking protein expressions within the mTOR pathway. Notice the bands for phosphorylated effectors, PRPS6 and P4EBP1. In the MHY1485 treated cohort, these markers fully recover from the suppressed teta baseline, confirming the pathway is active again. The elemental metabolic paralysis is bypassed. Even under conditions of severe copper deficiency, the biochemical machinery required to build white matter is back online. These fluorescent images and charts evaluate the impact on progenitor cells at embryonic day 18.5. The number of PDGFR alpha-positive early-stage oligodendrocytes shows a complete numerical recovery. Furthermore, the APC-positive quantification bars demonstrate successful maturation, proving the structural defect is repaired. To determine if this localized cellular rescue matters at a systemic scale, we transition forward in time to evaluate the 7-week-old adult mice. In the 3-chamber social tests, the MYY1485 treated adult mice exhibit a complete restoration of normal interaction times, freely engaging with novel social targets. This animated scatterplot isolates the definitive link between restored anatomy and behavior. The percentage of myelinated cortical area is on the x-axis, against social interaction time on the y-axis. The data reveals a strict correlation. As myelination restores, behavioral deficits vanish. The precise copper HIF1 BNIP3 mTOR signaling axis proves that the subtle balance of our trace elements dictates complex neurodevelopmental wiring and ultimately our social cognition.